The following is a demonstration of the Nextcode Health Clinical Sequence Analyzer. We will demonstrate the properties of the Clinical Sequence Analyzer using an actual case study that we solved using the tools you will see in the demo. In the study overview, we will begin by looking at the study subjects. For each study, one index case is chosen, often the first affected or the most representative case in the study. Others in the study are identified by their affection status and relationship to the index case. The study setup includes description of the phenotype. In this box, you can add phenotype signs and symptoms in order to generate gene lists that include known genes. For example, writing blindness adds a list of 61 genes. You can add your favorite genes by hand in the Add Candidate Genes menu. You can select one of several predefined gene lists, including NCI 37 and childhood recessive. You can click on the list name to see the gene details. You can also add your own gene list. Click Manage Gene Lists. You can manipulate previously added gene lists or upload a new gene list by clicking Choose File and finding a text file with gene symbols. Click on Upload. The gene list I uploaded is now available under User-Defined Gene Lists. Next, we can choose the Candidate Gene Analysis view, which analyzes the genes you have either added in gene lists or created with a phenotype signs and symptom, or added individually in the Candidate Genes menu. In this case, we have 3 candidates, but if we had many more than that, we might decide to filter the result. Filtering the results uses the frequency information. We will keep it at maximum 1%. Minimum read depth, we'll keep it at 10. We are only interested in exonic variants. We believe this is an autosomal recessive inheritance or compound heterozygote. We are interested in variant effect predictor consequences of high and moderate. Again, we can choose from the predefined or user-defined gene lists or add one gene at a time using the gene symbol view. We can choose to add paralogs of the genes selected and update the report. We can see that these actions definitively found the SLC52A2, the gene which turned out to be the disease gene, and including paralogs was the way this particular variant was discovered. There are other ways you can go about finding the same variant in the Clinical Sequence Analyzer, as I will demonstrate. Next, we can go to the Known Disease Variants view. The key thing to remember about this view is that it lists the known disease variants found in the index case. The index case is the reference for this analysis. The 10 results displayed here are 10 variants found in the index case that have previously been reported to cause disease. We can open filter parameters and filter based on the same criteria as before. Max frequency, minimum read depth, mode of inheritance, and variant effect predictor consequences. You can select or deselect 6 different HGMD variant types you are interested in. Update the report. This query returned no results, so we would move on. Next, we would go to the All Variants view. Again, the key thing to remember about this view is that it displays all the variants found in the index case. The index case is your frame of reference. In order to reduce the list of results to manually validate or eyeball, we again open filter parameters and filter based on the same criteria as before. Max frequency, minimum read depth, mode of inheritance, and variant effect predictor consequence. This time when we update the report, the the list has been reduced to 2 variants, including the variant we know is the disease gene in this case. You can open the vertical view here to get additional information. For the non-disease gene, we can open the vertical view and see that the mother has a call inconsistent with the index case at this position. In this case, none at all. Let's synchronize with the genome browser to see the BAM files. Confirm that the index case and her sister are homozygous and that their parents are heterozygous. And establish that the SLC52A2 riboflavin transporter is the disease gene. And now we wish to create a printable report. Because I have previously starred this variant, it appears in the Summary Findings view. I can choose to display any of 105 columns in the report. I can write a review comment in this box. Next, I click Generate Printable Report. This document summarizes the results in a concise manner. Look for other training videos at nextcodehealth.com/training videos.